You've seen that genetic engineering can fundamentally transform the very way that we perceive the world, but do we actually truly understand how to do that?
Dun, 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 dun. Dun, 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 dun. Dun, 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 dun. Dun, 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 dun. Dun, 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 dun.
Welcome, students, and all you curious folks out there. My name is Tarek, and today we're going to talk about the CRISPR technique. One of the most innovative tools with the greatest potential for humanity, without a doubt, is the technique known as CRISPR. This acronym stands for Clustered Regularly Interspaced Short Palindromic Repeats. In English, of course. Did that make it even harder to understand? I'll calmly explain what it means. It was created in 2012 by two researchers who developed a manual on how to use this technique in molecular biology, and because of that, they won the Nobel Prize in Chemistry in 2020, since this technique is so fantastic and important. In 2013, a Chinese researcher decided to use gene editing with the CRISPR technique inside eukaryotic cells. In other words, his goal was to make modifications to the DNA of cells that are very similar to human cells. That way, he would be able to change the phenotypic characteristics of these cells, which was a very important step. But what made this technique so important wasn't just the ability to edit, because we already knew how to do that, but rather the fact that it was a very simple, cheap, and fast technique. So a lot of researchers started working with this technique, and as a result, many things began to be discovered, including some not so good things, which led several countries to come together to discuss the issue. But we'll get to that and talk more about what it means later on. So let's explain what this technique is and how it works. In the 1980s, scientists found bacteria that could modify the nucleotides in DNA to prevent viral infections. Nucleotides are those famous little letters, A, T, C, and G, that we saw in the DNA lesson here on the channel. They stand for adenine, thymine, guanine, and phytosine. Here's what the bacteria did. When a virus entered its cell and injected its DNA, that viral DNA would attach itself to the bacterium's genetic material. The bacterium would then recognize this using pre-programmed DNA sequences it already had, called CRISPRs, and would create a specific RNA with the segment the virus had inserted there. This is the CRISPR RNA. This new RNA would bind to the part that the virus had inserted and signal to the bacterium what was wrong in its DNA. Then, the bacterium would create specialized enzymes to cut the genetic material. That way, the viral DNA segment would be removed from the bacterium's DNA, and the bacterium would be free from the viral infection. Then came the idea, what if we could create RNA molecules using the same technique that the bacterium used to remove the parts of viral DNA it didn't want? Except, we're going to manipulate this for any part of the DNA we want. So, if we know the nucleotide sequences we want to edit, we can create a specific CRISPR for that DNA sequence we want to modify. So, we use an enzyme called Cas9 to cut the target DNA, and that's it. You cut and replace that nucleotide sequence with one of your choosing, or you simply eliminate it. You literally create specific genetic modifications for whatever you want, no longer depending on the natural evolution of organisms. You create your own evolution by making these modifications. But what can we actually use these techniques for? Well, there are literally thousands of applications. From genetically modifying agricultural products to create resistance to pests, like we talked about in the banana lesson here on the channel, to changes in taste, shape, and a bunch of other things. We can also use it to cure genetic diseases by modifying defective genes, or even for genetic enhancement in living organisms, including humans. But then there's the dark side of this technique. The use of CRISPR for genetic editing in humans is something that's been heavily debated and is very close to becoming a reality. In 2015, a group of Chinese researchers decided to use this editing technique on human embryos to correct some mutations in a gene related to thalassemia. In 2017, another group published an article in Nature doing something similar with another group of genes that would cause heart problems in children. The idea is good, modify the genes in a child's embryo so that they aren't born with the disease. So far, nothing out of the ordinary. The problem is the precedent it sets. Genetic editing applies to diseases, but it also applies to any human trait. So, assuming this becomes normal, what's to stop fertility clinics from modifying an embryo to make it stronger, with fewer genetic problems, or even changing hair, skin, and eye color to whatever you want? This is a very complicated issue that needs to be discussed because it's both possible and foreseeable. In fact, a movie from 1997 had already warned about this possibility. That's why in 2017, a group of American scientists released a report establishing a series of rules for using this technique so that it's restricted to treating diseases and not for genetic enhancements. We also have to point out that it wasn't just this group of scientists who spoke out. In fact, there were several groups talking about the topic, including UNESCO, using the information and discussions on the subject, as well as deliberating on how this technique should be used in stem cells, which we also discussed in another lesson here on the channel. So we can say that this technique is an incredible tool that could revolutionize science and the way we deal with the world, from agriculture to medicine. However, if it's not used properly, it can become a powerful weapon against humans, so we need to be very careful. I hope you enjoyed the video. Don't forget to like and share, and remember that we have a new video every week, so subscribe to the channel and turn on the notification bell to always get the latest updates. Thank you so much.